Confused about what actually separates prokaryotic from eukaryotic cells or why cell theory even matters for the MCAT? These are foundational ideas that show up everywhere, from biochemistry passages to experimental questions. In this quick review, we'll break down the core principles of cell theory and give you a clear, test-ready way to distinguish cell types so you can recognize them instantly on exam day. Cell theory states that all living organisms are composed of cells. The cell is the basic unit of life, and all cells arise from pre-existing cells. This is one of the most foundational concepts you need for the MCAT because it frames how we think about all biological systems. There are three classic components. 1. All living things are made of one or more cells. 2. The cell is the smallest unit capable of carrying out life processes. And 3. Cells come from other cells through division. A helpful mnemonic is all, basic, come, or ABC of cell theory. On the MCAT, this often shows up in questions about growth, development, and disease. For example, cancer can be understood as a breakdown in controlled cell division, directly relating to the idea that new cells arise from existing ones. It also connects to reproduction topics like mitosis and meiosis, which are mechanisms that generate new cells. Understanding that the cell is the basic unit of life helps you interpret experimental data, such as when researchers study cell cultures to model entire organisms. If a question describes something alive but lacking cells, like a virus, that should raise a red flag, because viruses are not considered living under classic cell theory. This concept also ties into evolution, since all cells share common features like DNA and membranes, suggesting a common origin. Mastering this principle gives you a framework for nearly every biological system you will study for the MCAT. Prokaryotic cells lack membrane-bound organelles and a nucleus, while eukaryotic cells contain both. This distinction is one of the highest yield facts on the MCAT. Prokaryotes, which include bacteria and archaea, are simpler and smaller, typically around 1 to 5 micrometers, and their DNA is located in a nucleoid region rather than a true nucleus. Eukaryotes, including animal, plant, fungi, and protist cells, are larger and compartmentalized with organelles like the nucleus, mitochondria, and endoplasmic reticulum. A useful mnemonic is pro equals no, as in no nucleus, and EU equals true, meaning true nucleus. On the MCAT, you may be asked to identify cell types based on descriptions of structures. For instance, if a cell has ribosomes but no nucleus, it is prokaryotic. If it has mitochondria and a nuclear membrane, it is eukaryotic. This concept also connects to transcription and translation. In prokaryotes, these processes occur simultaneously because there is no nucleus separating them, whereas in eukaryotes, transcription happens in the nucleus and translation in the cytoplasm. Another common MCAT angle is antibiotic targeting, since many antibiotics exploit differences between prokaryotic and eukaryotic cells, such as ribosome size. Understanding these differences helps you interpret experimental setups, evolutionary relationships, and even medical treatments. Prokaryotic cells typically have a single circular chromosome and may contain plasmids, while eukaryotic cells have multiple linear chromosomes. This difference in genetic organization is highly testable on the MCAT and has important functional consequences. In prokaryotes, the circular DNA is located in the nucleoid region and is not enclosed by a membrane. They can also carry plasmids, which are small circular DNA molecules that often contain advantageous genes like antibiotic resistance. In contrast, eukaryotic cells store DNA in a membrane-bound nucleus, organized into multiple linear chromosomes wrapped around histone proteins. A helpful way to remember this is prokaryotes are simple circles, eukaryotes are complex lines. On the MCAT, This distinction often appears in questions about gene regulation, replication, or inheritance. For example, plasmids are commonly used in biotechnology and cloning experiments, which you may see in passage-based questions. Additionally, the presence of histones in eukaryotes connects to epigenetics and gene expression control. This topic also links to the cell cycle, since linear chromosomes require specialized structures like telomeres to protect their ends during replication. Understanding how DNA is structured in different cell types helps you predict how quickly cells replicate, how genes are regulated, and how mutations may spread. Eukaryotic cells are compartmentalized into membrane-bound organelles that allow specialized functions and increased efficiency. This is a key advantage of eukaryotic cells over prokaryotic cells, and a frequent MCAT testing point. Organelles like the nucleus, mitochondria, lysosomes, and endoplasmic reticulum each have distinct roles. For example, the mitochondria generate ATP through oxidative phosphorylation, while the rough endoplasmic reticulum is involved in protein synthesis. This compartmentalization allows incompatible processes to occur simultaneously without interfering with each other. A helpful analogy is thinking of a eukaryotic cell as a factory with different departments, each specialized for a task. On the MCAT, you might be asked to match organelles with their functions or predict what happens if an organelle is damaged. For instance, if lysosomes fail, waste accumulates, leading to cellular dysfunction. This concept also connects to protein targeting and trafficking, where signal sequences direct proteins to specific organelles. Additionally, compartmentalization plays a role in metabolic pathways, allowing gradients and controlled environments, such as the proton gradient in mitochondria. Understanding this organization helps you interpret complex biological processes and experimental data involving cellular function. Cell size and surface area to volume ratio limit cell growth and influence whether organisms are unicellular or multicellular. This concept is essential for understanding why cells remain small and why complex organisms are made of many cells rather than one giant cell. As a cell grows, its volume increases faster than its surface area, which reduces the efficiency of nutrient uptake and waste removal. A useful mnemonic is volume grows faster than surface, reminding you of this imbalance. On the MCAT, you may encounter questions that require you to reason about diffusion rates, or why certain cells have adaptations like microvilli to increase surface area. For example, intestinal epithelial cells have microvilli to maximize absorption. This principle also explains why cells divide rather than continuously grow larger, connecting directly to the cell cycle and mitosis. In multicellular organisms, specialization of cells allows different tissues to perform specific functions efficiently, overcoming the limitations imposed by surface area to volume constraints. This topic also ties into physiology, such as gas exchange in the lungs, where alveoli maximize surface area. Understanding this concept helps you explain structural adaptations and predict how changes in cell size can affect function, a common reasoning skill tested on the MCAT.